Today's topic for discussion is topic of plant pathology, and today's topic for discussion is citrus canker disease. Citrus, it is a very common fruit which is everyday used in kitchens of India and abroad, and the citrus plant that is affected by a bacterial disease, and that disease is known as citrus canker disease. Citrus canker, it is a bacterial disease, just I told you. It is cosmopolitan in occurrence. That means it occurs throughout the world where citrus is grown. It is a very serious disease. It causes heavy losses, and the disease causes serious damage in India, China, Japan and Java. The citrus plant, it is a perennial plant, and once we grow it for its fruits and several years it remains in field, and once when it gets infected, that infection is retained on the plant. The pathogen incites severe canker disease in number of citrus species on the stem. That means the citrus canker symptoms occur on stem, leaves, fruits, except root system. The disease occurs most of the species or most of the varieties of the citrus plant. The most susceptible varieties that shows more infection, and even this citrus canker pathogen attacks sweet orange and grapefruit, etc. Let us see the type of symptoms occur on the plant. Symptom, it is the expression of the disease. After interaction of host and pathogen, something new type of expression is developed on the infected plant, that is known as symptom. Symptom, it is a result of interaction of pathogen and host. Let us see the symptoms of citrus canker disease. Citrus canker disease, it produces somewhat raised pustules or lesions on the surface of the leaf, mostly on the lower surface of the leaf and on the fruit skin. In severe cases of disease development, such a type of brown raised corky spots are developed on stem and branches also. And the trees are, however, not commonly killed, but infection directly affect the yield. The lesions or the spots which develop on the leaf, they first appear on the lower surface as a small, slightly raised, very small spot-like structure, and these are yellowish brown in color. These structures later on turn white or greyish and finally rupture, and this spot is corky in appearance. It is a spot caused by a bacterial pathogen. The older lesions, many older lesions, lesions means spot, they mix, and most of the area of leaf is covered. Sometimes purplish in color. These spots are purplish in color, purple colored. The necrotic brownish canker region, that means the canker region where the spot is developed, that consists of dead tissue. Hence it is called necrotic spot or necrotic brownish canker region, and that is surrounded by yellowish brown margin, and that margin is distinct watery yellow hollow structure. The yellow hollow region is free from the pathogen. The cankerous region contains the pathogen in millions. Millions of bacterial pathogen occur in each raised spot or corky spot of citrus canker, which is caused by Xanthomonas citri. And there is in this spot, as per some plant pathologists, they have reported that malic acid is present, and malic acid in the hollow region due to increased respiration in the region. The lesions on the twig are usually irregular in form, irregular in shape. The lesions or corky spots on the fruit are similar to those of the leaves but lack the yellow hollow. This pathogen, when it attack to the fruits, even it attack to the fruit, but this infection remains that this infection remains up to the skin only. Fruit skin is that shows infection, and such fruits it becomes difficult to farmer to sell. But this infection remains up to the skin only because it never enters into the pulp of the fruit. Okay, next, causal organism. The disease citrus canker, it is a bacterial disease, and name of the bacterium which causes citrus canker disease, it is known as Xanthomonas citri. Now this pathogen, it is called as Xanthomonas compactus form species citri. Okay, it is a bacterium. It consists of a rod-shaped cell. The size of this bacterium is 1.5 to 2.0 into 0.5 to 0.75 mu. This is a rod-shaped bacterium. It has a flagella, hence it is a motile bacterium. And this flagellum, it has a single flagellum which is situated at the polar region of the cell, bacterial cell, and such a single-celled bacterium that is described as monotrichous bacterium. It lacks endospore formation. This bacterium do not produce endospore in adverse environmental conditions. It is a gram-negative bacterium and it is obligate parasite. Why it is called obligate parasite? Because it requires living host plant for its survival, hence it is obligate parasite. And this bacterium, it is also called as aerobic bacterium because it requires oxygen for its growth and survival. And this bacterial cell that is enveloped by a mucilaginous capsule-like structure, that is for protection, and it forms a chain. This bacterium forms a chain or it occurs in the chain form. The climatic factor which favor the disease are mild temperature and wet weather. In such environmental conditions, the pathogen multiply rapidly, and whenever it comes in contact with the host plant, it causes citrus canker disease. And the suitable temperature for the growth of this Xanthomonas compactus, it is a pathogen which causes citrus canker disease, is the temperature is 20 degree to 30 degree Celsius temperature is favorable. During such a temperature, it can grow normally. It can cause disease. Next, this is all about causal organism. Let us see the disease cycle. Infection takes place through stomata. The pathogenic organism, they say that the pathogen is a bacterial pathogen and it is an obligate parasite that requires only a living host plant. And that the citrus plant, it is a perennial plant, and the plant remains in the field for several years, and the pathogen remains on the infected plant for several years because the host is perennial. And through the wind or through the insect visits, the pathogenic organism is transferred from one place to another. This is the main source of the pathogen. And what happened? The disease is not soil borne. It do not occur in soil because it is an obligate parasite. It requires only and only living host plant for its survival. And the pathogen perinates in the old lesions on the twigs still attached to the host plant. From there it is carried by, or it is transferred from by insects or raindrops to the new localities or to the new host plant. Even the same plant get infected, and even we, whenever during harvest of the lemon, we visit, workers visit the plant and they collect the lemons, and at that time also this pathogen transfer from one host to another. These are different agencies of transmission of disease from infected to healthy plant. Let us see details about control measures. As the pathogen is obligate parasite and even the host is a perennial plant, eradication is the method. For some extent we can reduce the disease intensity. The disease is controlled by eradication of diseased tree. And in eradication, what we have to do? We have to take a sterile polythene bag, and with the help very carefully we have to remove the infected leaves from the infected plant very carefully, and we have to take it in that sterile bag. When we have to dug it, or we have to burn it far away from the infected plant. In this way we can manage the disease, we can reduce the disease intensity. Next method for the management of the disease is pruning. The infected trees may be carried by removing the diseased foliage and branches with pruning scissors, and then spraying the trees with 1% Bordeaux mixture at regular intervals. We can manage the disease. And we have to use disease-free nursery stocks for sowing purpose. And every time we have to collect all the defoliated leaves or the leaves which fell off on the ground because of infection, that we have to remove from that field. We have to keep the field always clean. And we can spray. There are different fungicides available in market, recommended fungicide and recommended doses we have to spray for the management of the disease. There are several antibiotics used by the plant pathologist for the management of the disease. Rangaswamy, he has reported streptomycin sulfate and phonomycin for the management of the disease. And recently there are several different varieties of the citrus has been developed by plant breeders, and those are resistant to the disease.